This training video guides you through the steps to successfully conduct a test using Neogen's Reveal Q+ for Don and the AccuScan PRO reader. This presentation serves only as a visual guide to the written materials supplied with the test kit. To ensure accuracy in the performance of this test, please read and follow the test's written instructions in their entirety. Each kit contains enough sets to test 25 samples. The kit includes test strips, Reveal sample cups, sample dilution cups, sample diluent, and the QR code. To conduct the test, you also need the following: sample collection cups with lids, an agri-grind or bun grinder, a scale capable of weighing 5 to 50 grams, accurate to plus or minus 1/10 gram, a timer, a Reveal sample cup rack, the Reveal AccuScan PRO reader, dispensing pump or graduated cylinder, filter syringes or Whatman No. 1 filter papers, sample dilution tubes with caps, a 100 microliter pipetter, a 500 or 1000 microliter pipetter and tips, and distilled or deionized water. Remember to always store test strips in the Stay Dry tube at room temperature. Do not freeze the test strips. Treat all used liquids and labware as if contaminated with Don. Neogen recommends gloves and other protective apparel, although they're not required during testing. Be sure to follow your company's policies. Avoid cross-contamination by using clean extraction supplies for each sample. Neogen's AccuScan PRO system consists of a lateral flow test reader and data management software that interprets Neogen's Reveal Q+ test strips for mycotoxins. The system provides an easy method to objectively read, store, and analyze results. Plug in the AccuScan PRO system to turn the reader on. Following a 3-minute startup and self-calibration, the reader arrives at the home screen, which displays 4 icons: Run Test, QR code, History, and Settings. Prior to running a sample, you must enter the lot-specific QR code. To do this, place the disposable QR code contained in the kit into the QR code cartridge. Select the Scan QR code icon from the home screen and place the QR code cartridge in the reader. The specific information is automatically downloaded into the reader. Alternatively, you can manually enter the QR's 39-digit code. For instructions on how to do this, see the AccuScan PRO user manual. It is important to scan the QR code for each new kit lot. To prepare the sample, begin by gathering all of the materials required for the test. The sample to be tested should be collected according to accepted sampling techniques. See the GIPSA sampling protocol or contact your Neogen representative. Obtain a representative sample by grinding a minimum of 100 grams of sample so that at least 75% of the ground material passes through a 20 mesh sieve. The particle size should be about the same as instant coffee. The sample is extracted with a ratio of 1 part sample to 10 parts distilled or deionized water. For this example, we'll use 10 grams of ground wheat with 100 milliliters of distilled water. The USDA GIPSA approved method uses 50 grams with 500 milliliters of distilled or deionized water. Vigorously shake by hand or mechanical means for 3 minutes. Alternatively, you can pipette 1 milliliter of the sample into a 1.5 milliliter microcentrifuge tube and centrifuge for 1 minute using a microcentrifuge. Or you can blend the sample for 1 minute. Allow the sample to settle and filter with a filter syringe or Whatman No. 1 filter paper. For each sample you're testing, place sample dilution tubes and Reveal sample cups in a sample cup rack. Add 1,000 microliters of sample diluent into each red sample dilution cup. Then add 100 microliters of the sample extract. Mix up and down with the pipette at least 5 times. Transfer 100 microliters diluted extract to a Reveal sample cup. Use a new pipette tip for each sample. It is important to make sure the pipette tip is fully charged with liquid and doesn't have any air bubbles prior to dispensing the liquid into the cup. Place a Reveal Q+ for Don test strip into each sample cup with the sample end down. Allow the strip to develop in the sample cup for 3 minutes. During this time, prepare the AccuScan PRO reader to read your results when the incubation is complete. To do this, touch the Run Test icon on the home screen. The test category displays in the first box, and the test type displays in the second box. Select mycotoxins. Select Don Q+, then touch the green check mark to confirm the selection. If entering a sample ID, type it in and press the green check mark to continue. Strips may also be tested without a sample ID. Following the 3-minute incubation, remove the strip and insert it into the R-labeled strip cartridge. Insert the cartridge containing the strip into the slot on the lower left portion of the reader, square end first. The reader automatically begins analyzing the strip. It's important to note that readings must be conducted within 1 minute of the completion of the 3-minute incubation. Readings conducted after 4 minutes may be inaccurate due to overdevelopment of the device. Strips must be read using the Reveal AccuScan PRO reader. The screen displays an exact parts per million result. Touch the screen to view additional test information, such as test line and control line ratios. To print a report, select the print icon to send the report to an attached printer. To begin a new test, select the New Test icon to return to the test setup screen. The previously selected test is saved as the test type. Results can be exported into the AccuScan Data Manager software through a USB key or by data transfer via Ethernet connection. If you have any questions regarding the use of the AccuScan PRO reader, please contact Neogen. Manuals are also available. Neogen now has a full line of Reveal Q+ test kits, including aflatoxin, aflatoxin green, Don, zearalenone, fumonisin, ochratoxin, and T2-HT2. We have kit inserts and step-by-step pictured procedure brochures for all Reveal Q+ kits. Please contact Neogen for more details on our full line of Q+ test kits.